Before CRISPR-based methods took center stage in the genome editing field, other bio-inspired tools such as zinc finger nucleases and Cre-lox system recombinases made genomic engineering possible. Some researchers are now looking back to these foundational technologies to improve upon and one-up the latest gene editing techniques. Welcome to The Scientist Speaks, a podcast produced by the Scientist Creative Services team. Our podcast is by scientists and for scientists. We bring you the stories behind newsworthy molecular biology research. This episode is brought to you by Oxford Nanopore. Oxford Nanopore offers industry-leading sequencing solutions across the whole spectrum of biopharmaceutical research and development, from initial target identification and biomarker discovery to cell line and viral vector engineering, all the way through to cell, gene, and RNA therapy drug development. In this month's episode, Deanna MacNeil from The Scientist spoke with Frank Buchholz, professor and head of the Medical Systems Biology Translational Research Group at the Technical University of Dresden's University Cancer Center, to explore how his laboratory designs programmable recombinases for high-fidelity gene editing.
Over the past several decades, advances in genetic engineering have allowed scientists to target and modify specific locations in the genome, with enzymes that cleave, weave, and repair DNA. Recombinases are one type of specialized enzyme that researchers have used to precisely cut and rejoin DNA at specific target sequences, manipulating the genome in highly controlled ways. Frank Buchholz's journey in genetic engineering began while working with one of the earliest recombinase-based gene editing tools, called the Cre-lox P system. When he launched his own laboratory, first at the Max Planck Institute and later at TU Dresden, he remained captivated by the idea that molecular tools like recombinases could one day unlock the ability to precisely rewrite the genetic code and open new treatment avenues for genetic diseases.
I've been always interested in genetic engineering. When I did my PhD more than 25 years ago now, the latest tools that became available were the Cre-lox P system, which is a bacteriophage protein that was repurposed that can recombine at particular sites called LOXP. And this was a breakthrough at the time because this allowed for the first time to do conditional knockouts in mice, for instance, so that one could delete a gene particularly only in a specific organ, for instance. And this was fascinating me that this is possible to do. And I decided that that was something that really interested me. And I got more involved and more interested in this group of proteins to see how they can be used and potentially be further developed to make them even more useful. Well, I wasn't the only one excited about being able to manipulate the genome of living organisms. And in fact, before all of this happened, the human genome and other genomes were sequenced, and there was a lot of interest in trying to say, now we will understand how life works and how the systems work. And nobody was thinking that being able to write in genomes would come before understanding how it works. Everybody was surprised to find that with such precision and such efficiencies, we are now able in organisms to basically change the genetic code so that we can start thinking about using these things for therapies.
Although Buchholz's journey began with recombinases, the genome editing field's origin story starts with a different enzyme family, called nucleases, which cut DNA but do not repair it. Investigations in the early 1990s showed that scientists could use nucleases to target and cut specific sites in the DNA, which the cells then repaired via host DNA damage repair pathways. This established the concept of genome editing using double-strand break generating nucleases, including homing endonucleases, zinc finger nucleases, and talons, each of which paved the way for the programmable nucleases that support the genetic engineering research field today. These tools allowed scientists to cut DNA with precision, opening up new avenues for functional genomics and, eventually, therapeutic applications. Then came a seismic shift, as the discovery of CRISPR and its applications in genome editing redefined what was possible in the field. CRISPR-Cas systems use RNA-guided nucleases that synthetic biologists modify and direct to orchestrate targeted edits almost anywhere in the genome. CRISPR rapidly became the centerpiece of modern gene editing, accelerating research across biology and medicine. But while CRISPR has transformed the landscape, it's not without its challenges.
When you have a nuclease, you introduce a double-strand break, then the cell has to somehow repair this DNA double-strand break. And the cell has different ways to do that. So it's unpredictable, basically, how each individual cell is going to do it, with the possibility that each individual cell does it slightly different. And of course, that makes it difficult to predict whether this is useful or this is potentially also dangerous. CRISPR nucleases make a double-strand break. More sophisticated systems like base editors or prime editors don't induce double-strand breaks and therefore are considered safer, but still also they do require the DNA repair pathways to finish the reaction. They still create a nick, and they still then need the host repair mechanisms to repair these lesions. And potentially also then they might suffer from this, that the cell is not repairing every cell exactly the same way.
Buchholz's research positions recombinases as alternatives to the nuclease-based technologies that dominate today's gene editing toolkit. For example, the Cre-lox P system, that was discovered more than 25 years ago, remains a very useful tool that scientists employ for conditional gene knockouts. But to use the Cre recombinase in this manner, researchers first must integrate the specific LOXP DNA sequence into the genome at the target site, which is a roadblock to therapeutic applications. As a solution, Buchholz and his team now engineer programmable recombinases that recognize and act upon naturally occurring target sequences. These enzymes blend nuclease and recombinase domains to create a novel enzyme that recognizes a specific DNA sequence and recombines it, doing so with high fidelity and without involving cellular DNA repair pathways.
The idea basically was, if we would just be able to reprogram the recombinases, then we would have an ideal tool. So we start off with a very mild modification of the LOXP site, of the natural sequence of that recombinase. We then lose a lot of activity of the natural enzymes. We mutate the coding sequence and then select out variants which have higher activity on the new site. Then we move closer to the target site that we want the enzyme finally to recombine. And this way, it's a stepwise process of directed evolution to then have an enzyme which finally recombines a therapeutically interesting target sequence. This has been the bread and butter of how we're making new recombinases up until recently.
Buchholz and his team have used this stepwise in vitro approach to evolve more than 100 different Cre-based recombinases that act on new target sites. Although this iterative method has been effective, it is a slow process that depends on analyzing each recombinase variant's coding sequence to understand which changes contribute to the new recombinase's activity and specificity. Here too, technological advances are playing a role in improving a foundational method. Buchholz uses deep sequencing and deep learning algorithms to accelerate directed evolution and predict which changes they should engineer into an enzyme so that it will successfully edit a new target.
We developed a protocol where we use long-read nanopore sequencing. We're getting for a library of enzymes quantitative information now, where we know this amino acid sequence of the recombinase has that activity on the wanted target site, but at the same time, we can also query many different sequences that are related. So we also get an off-target profile for each sequence. So we have very detailed information on amino acid sequence from the protein and its on and off-target activities. And this is fed into the algorithm to then make predictions on how a new sequence would behave. It helps us to basically bootstrap so that we make sub-libraries that predicts what we have to change and where. So it helps in accelerating the whole process. We're still working also, of course, to continuously feed in new data to make these predictions more accurate and more reliable.
As for any genome editing technology, it's crucial that the new recombinases do not modify any closely related or off-target sites in the genome. In addition to their deep sequencing approach to query off-targets, Buchholz has turned to zinc finger nucleases, another veteran technique in the genome editing toolkit, to boost the specificity of the programmable recombinases that he and his team are developing. In recent work published in Nature Biotechnology, the researchers fused Cre-type recombinases to zinc finger DNA binding domains, crafting programmable recombinases whose activity depends on DNA binding, and thus improving their specificity.
We first did NNC terminal fusions, and we saw that we can indeed boost activity and also boost for this specificity. But luckily, we also decided to test what happens if we put the zinc finger in the middle of the recombinase coding sequence. And we almost threw this idea into the bin, because when we then first tested these enzymes, we saw that they had absolutely no activity. Luckily, we also tested these enzymes on target sites where we placed the recognition sequence of the zinc finger at varying distances to the target site of the recombinase. And what we found, surprisingly, was that if the distance was the right distance between zinc finger binding site and recombinase recognition target site, we could totally recover the activity of this enzyme. So now we made a recombinase that was conditioned on the zinc finger to first bind its target site, and this had to be at the right distance to the target site for the recombinase. Only then we could recover activity of the recombinase. And this, of course, gives us a lot more specificity, but also gives us a lot of freedom. We can uncover the recombinase specificity from DNA binding. And this was very important because now we don't have to spend so much time on making the recombinase so specific. Indeed, we went back to an enzyme that we once had made in the laboratory that was very unspecific. That's the enzyme that we called RECFLEX, which basically is very flexible. It's a very toxic enzyme that we had evolved. It recombines many, many sequences, and so it's basically useless. And we then said, maybe we can now tame this really unspecific recombinase if we stick the zinc finger domain in the middle of this enzyme. And indeed, we saw that this enzyme totally lost activity on all of the target sites that it had, unless we placed the zinc finger binding sites again next to the target site of the recombinase. Basically, it shows you that in the lab, you should never throw anything away because you never know what it might be useful for in the end.
By revisiting RECFLEX with their new zinc finger fusion method, Buchholz discovered a different way to design programmable recombinases that somewhat mirrors how CRISPR-Cas9 remains inactive until guided to the right target site by its RNA component. The recombinase is inactive unless its DNA binding domain has found its site, and only then the recombination activity can take place. Building on this, the researchers demonstrated that the system works not just with zinc finger domains, but also with other DNA binding domains. The team also tested their approach across a range of recombinases and created custom DNA binding domains with their sights set on therapeutic targets. Their hope is that recombinase-based gene editing will evolve into a new class of curative medicine, treating the root causes of disease, not just the symptoms.
The first recombinase we actually made is a recombinase that can excise the integrated HIV provirus from infected cells. And this is the project that's also the furthest in terms of translation. So we have obtained funding to perform a clinical trial. The vectors have been produced in GMP quality and so on. All the preclinical work is virtually finished. And so we are hoping to start a clinical trial soon. We've made an enzyme that can correct the genetic inversion that is underlying hemophilia A. We also aim to correct MECP2 duplication syndrome. This is a disease where a piece of DNA is duplicated, and we intend to make a recombinase that can delete this duplication. I hope that the technology has established itself and that first clinical trials give hopefully positive results, that this can help patients to offer a new type of medicine which offers cures rather than treating symptoms, because that's the beauty, of course, of all of these technologies, that you really get to the root of the problem of a disease and you repair the underlying problem. So we think that the technology is really a major advance to the field. In the end, I think it will help to develop, especially for large edits, fluent writing in genomes, to also finally understand the grammar of life, because I think It will be central to really understand how the DNA encodes life, how to really get at the detailed steps of how this is working. We need to be able to make precise changes in the genome to then learn how the whole system works. I think this is essential. So these are the future applications that I see that excite me most.
Thank you for listening to The Scientist Speaks. This episode is produced by the Creative Services team for The Scientist and narrated by Deanna MacNeil. Thanks again to Oxford Nanopore for sponsoring this episode. Please join us again in October as we learn how researchers harness artificial intelligence to fight infectious diseases. To keep up to date with this podcast, follow The Scientist on social media and subscribe to The Scientist Speaks wherever you get your podcasts.